Hi, thank you for joining me today. My name is Lindsay Rickershauser, and I am part of the Cheminformatics Technologies team within MilliporeSigma, representing our retrosynthetic design software, Synthia. Today I'm here to tell you about our novel and in silico pathway design that has been validated at the bench. Virtually every day, a chemist is tasked with the objective of synthesizing a new target molecule. This sounds like a simple request, but we as chemists know that this is neither simple nor easy in a lot of cases to get to a final successful outcome, especially when additional constraints begin to get added to that simple request, such as finding a shorter, enantioselective route that utilizes cheap starting materials and avoids hazardous reactions. The complex web of possibilities gets even more complex with each of these new constraints that we add to our retrosynthetic design. If we attempt to solve this by hand, it could take days, maybe even weeks, to find a suitable solution. However, put into the hands of a retrosynthetic design software, this problem can potentially be solved in just minutes. Synthia is a highly customizable retrosynthetic design software that can help a scientist to begin to distill down to exactly what matters to them within the complicated and expansive graph of possibilities for their targets. Within the software, a scientist can input their target and describe their needs for this retrosynthesis. Once those constraints are input, this allows the algorithms to then simplify that complex web of possibilities to focus on, find, and rank the pathways that matter to you as the end user. Some of the aspects of a search that an end user can customize are things such as the number of steps in that synthetic pathway, the cost constraints of any building blocks that you may need to buy for that synthesis, any protecting group requirements that you may have, for example, if you need a protecting group-free synthesis. You can even avoid toxic or undesirable intermediates or building blocks, catalysts or reagents, even entire chemistry reaction classes. You can even select within Synthia the building blocks databases that you would like it to plan synthetic pathways back to within your search so that it hits the right criteria for this particular project. So how does Synthia know what reactions are possible for your particular target? We have very explicit rules within our database that allow Synthia to define that search tree. Every single rule constitutes a chemical reaction that has been published somewhere in the literature that you may know from your schooling or your time at the bench. However, each rule that exists within the database that gives Synthia that context to be able to expand that graph of possibilities is hand-coded by an expert-level chemist to specifically and systematically account for things such as incompatible functional groups, protecting group requirements within that pathway, stereo and regiochemistry. So where you need an enantioselective synthesis, Synthia is able to give that to you. And it even comes back and stops at those commercially available and known starting materials, allowing Synthia to propose viable pathways to novel molecules, not just what has been published within the literature previously. And so once it identifies a reaction as possible, it starts to look at whether it is viable, taking all of that information and context from the rules that we're creating and applying it to any reaction that is proposed by those algorithms within Synthia. So as a reaction is being proposed, it looks and excludes that core of that reaction to evaluate if there are any protecting groups that could be required for that step in the synthesis, or any incompatible functional groups that would disallow that reaction from going. And so if no protecting groups are required, it would return a list of any incompatible functional groups within that particular reaction. And if they existed, it would actually filter that out as a viable option from that result set for your product. If a protecting group is required, it would select a protecting group that would be compatible with those reaction conditions, and then it would return it with that reaction as it's being proposed in your result set. Synthia continues to traverse the graph of possibilities until it has found full pathways that stop at commercially available or known starting materials, and throughout the course of each search compiles hundreds, if not thousands, of potential results for a target molecule. As part of its search strategy, Synthia evaluates all of the retrosynthetic cuts and possibilities at each level, ranking and scoring those particular retrosynthetic cuts against the viability and the search parameters put forth by the end users, only keeping those best solutions and continuing to go deeper into that retrosynthesis, all of the while eliminating those non-viable reactions that it identifies, continuing on until it gets to those terminal stopping points, which would be those commercially available or known building blocks. And so with all of the results that it compiles throughout the course of the search, the question then becomes exactly how to rank those results according to that end user's needs. And that is what the scoring function is able to do to help Synthia prioritize and find and rank the best results according to that end user. Because you can imagine that what could be the best overall pathway for one chemist may or may not be what another chemist would select according to their needs. For example, one scientist may be looking for the shortest overall pathway, regardless of any pricing of those starting materials that could be required, where another chemist may be looking for a route that avoids palladium, and even a third may be looking for a synthesis that utilizes chiral pulled building blocks so that they don't have to worry about enantioselectivity issues. These are all customizations a chemist can make in Synthia's scoring functions when setting up a new search, so that they can tailor each unique search according to their needs. All of these selections will shape exactly how the algorithm searched the graph of possibilities and ultimately affect how each pathway found will be scored and ranked for that end user to analyze. So as you can imagine, we were highly excited about this technology when we saw it, but up until that point in early 2016, there had been no experimental proof that you could actually utilize the software to plan a synthetic route and then execute it successfully at the bench. And so we engaged in an experiment to do just that, where we identified 7 molecules of very high financial interest to our business, some molecules suffering from low yield and high cost. One molecule in particular, the only published synthesis had been under patent, and so for legal reasons we had avoided synthesizing it. And some other molecules had been novel, never published in the literature before, and the routes identified internally had been deemed too risky. So 6 chemists actually then traveled to the Polish Academy of Sciences in Warsaw, Poland to meet Professor Bartosz Drabowski and his team of graduate students that have created the software. We participated in a face-to-face training on the software, and we input our molecules to begin designing our synthetic routes. It was very important that the chemists actually executing the work at the bench were the ones planning those routes, because not only did we know what had been done in the space for those particular molecules, we also knew the constraints of our own facilities. So where we needed to avoid any particular reactions due to safety concerns, we could do that. Or where we wanted to find or promote pathways that contained building blocks that we had in stock or reactions that we wanted to pursue, we could do that as well. We did have rules associated with this experiment, so we wanted to mimic the everyday pressures that a chemist is under in terms of time. And so we needed to pick pathways that contained commercially available starting materials in stock and ready to ship, because once we got back to the bench, we had 8 weeks to execute on that synthesis. No changes were allowed to that route, so where the software proposed a Suzuki coupling followed by an amidation reaction, we had to execute those steps in sequence. However, where the software is great about giving a starting set of reaction conditions, it's not necessarily going to be able to give the best catalyst and ligand combination to affect the highest yield. So we fully expected some optimization in the process. While I don't have time to talk about all of this validation experiment, I do want to concentrate on one particular molecule, alpha-hydroxyetizolam. So this is a metabolite of etizolam, which is a drug that's used to treat depression. At the time that we were looking at this molecule, it was novel. There was no literature precedent to actually synthesize it. We had identified a route internally for this molecule. However, we had deemed it very high risk due to the installation of the hydroxyethyl group being done at a late stage in the synthesis. And so we had determined that a significant amount of R&D could be required because it was very high risk that we could fail in late stages of this synthesis. And so for that reason, that particular route had been sidelined. But when this collaboration opportunity came about, we thought this would be a great way to challenge the software to find an alternative, more robust route. And so in quickly looking at the results, Synthia will give you a graphical output of results, with the circular nodes being the starting materials, intermediates, and products of those reactions, and the diamonds being the actual reactions being proposed. You'll notice the color coding in the legend on the screen, with the yellow being the target that you search, the pink being commercially available, purple being unknown in Synthia, and those blue halos indicating that a protecting group would be required for that step in the synthesis. So this is the actual synthesis that Synthia proposed for alpha hydroxyetizolam. It started with a 3-component Jawald reaction, which was very important. The chemists actually chose this route for a very particular reason, so that they would know in the very first step of this synthesis whether the installation of that hydroxyethyl group was going to succeed or fail. And indeed, it did succeed. And so in just a few more steps, we were able to build that benzodiazepine ring, followed by that triazole ring construction, and a final deep protection enabled us to synthesize alpha hydroxyetizolam for the very first time in a 3.2% overall yield and 8 steps as proposed by Synthia. While this is a rather modest yield, it was access to this novel new product for the very first time, and we were able to also actually access additional analogs in that same year as new products in our portfolio by applying this particular route. It also represented a 40% cost savings based on our original proposed route that contained R&D, which was a huge impact to the project in and of itself. As we set out on this experiment, what we had hoped for was a 50% success rate, and what we actually achieved was unprecedented. Every single molecule that was put to the test in Synthia was able to be synthesized at the bench utilizing the software's pathway, and not only that, but the chemists were able to achieve significant cost and yield savings as compared to the previously executed in-house routes, demonstrating that even a novice software user could obtain very impactful results from the software quite rapidly. I'd like to highlight 2 additional molecules from this experiment. The first is THCAA. With the legalization of marijuana in the food and safety testing market, the demand for THCAA had increased at a rate that we were unable to keep material on the shelves. Our in-house route was laborious and generated many impurities such that the team could not manufacture high enough quantities. With the help of Synthia, we were able to cut the number of steps in half, quadrupling the yield and synthesizing higher quantities than we had ever been able to access, ultimately creating a 62% cost savings on the product. This was so impactful that this was in fact the only molecule that we did not end up publishing with our results in Chem in 2018. We retained this more efficient route as trade secret. In addition, I'd like to highlight hydroxylorazidone. At the time we were looking at this molecule, the only published route for it was patented, and so for legal reasons we had avoided actually synthesizing this molecule. But with the help of Synthia, we were able to find unique disconnections outside of that patented route so that we were able to manufacture this as a new product in our clinical and diagnostic testing portfolio. As a direct result of this successful experiment, Synthia was the very first retrosynthetic design software that was successfully validated at the bench on non-trivial targets. As a direct result, Synthia was then acquired by MilliporeSigma in late 2017. The software was formerly known as Chematica, which was created by Bartosz Jabowski and his team of graduate students over the course of more than 15 years of research. We've continued to develop and enhance the tool since then, and in 2018 rebranded from Chematica to Synthia, and in 2019 we launched our web application version of the product that you see today. Over the last few years, we have continued to innovate and develop Synthia such that it can continue to think more strategically like a chemist. In addition to the expert-coded rules, there exist within Synthia many different filters and functionalities to enable it to find efficient, elegant retrosynthetic solutions to very complex molecules. One of these functionalities is a filter that exists within the tool called strategies. Formally, these are 2-step reaction sequences that, when employed, allow the algorithms within Synthia to look 2 steps ahead in a retrosynthesis to find efficiencies in building that molecule that would otherwise be non-intuitive if looking only 1 step at a time. In early 2020, Professor Jabrowski and his team published the computer-driven discovery of large numbers of these two-step reaction sequences called tactical combinations, approximately 4.85 million, the vast majority of which had no previous literature precedent and had never been cataloged before. Up until this time, there were only approximately 500 cataloged two-step reaction sequences. I'd like to give you a few quick examples of these computer-discovered strategies. The first is example A, in which you have an exotrig radical cyclization followed by a tandem Birch reduction and hydrolysis of enol ethers, enabling the construction of a cis tricyclic ring system, which is found in triquinine sesquiterpenes. The second is example C, which you have an enantioselective rearrangement of enol carbonates to create a quaternary carbon center. The subsequent isomerization of that terminal alkene to an internal alkene forms a structural motif that can be further utilized in the synthesis of alkaloids. Finally, example H is the strategy based on a cationic cyclization of an epoxy alkane and the reduction of that furan. This is a combination of reactions that constructs a trialicyclic motif with a diterpenoid moiety that's present in numerous natural products such as linosterol and mercuric acid. All of these newly discovered strategies were then incorporated into Synthia in late 2018. We were further able to validate these new strategies that had been incorporated into Synthia with the execution of the synthesis of Emperonine. Once the new database of strategies was incorporated into Synthia, the team utilized the software to find a more concise route for Emperonine, a platelet aggregation inhibitor that had been published previously within the literature. By simply inputting their target and activating strategies along with Synthia's default search conditions, Synthia rather quickly returned several pathways that were shorter than the published routes and offered unique disconnections as to compare to what was previously published. Analyzing results within Synthia was rather quick and easy by clicking on any molecule or reaction nodes to find more information, such as the name of that reaction, typical reaction conditions, even links to the illustrative references in the literature. Any reactions where protecting groups may be required, Synthia will also give protecting group information along with suggesting protecting groups that will be compatible with those reaction conditions. Utilizing the filtering options also allowed the team to look at the most diverse reactions pathways that have been proposed within that result set and allow them to open up and take a look at the molecules as well instead of looking at that graphical node format to see how those pathways were being stitched together. Finally, if the chemists wanted to look at all of the 50 pathways being proposed for their molecule, they could take a look at the graph mode, which would enable them to identify the diversity in the last step reactions being proposed, identify common intermediates being proposed within the last step of that synthesis, and even find commercially available building blocks for the pathways they wanted to analyze further. Ultimately, the team identified the top scoring pathway within Synthia's results for Emperonine and were able to export that synthesis via PDF to get started with their benchtop execution. Up to this point, the shortest reported synthesis of Emperonine had been 8 linear steps as reported by Shattuck et al. In 2001. Synthia, however, had proposed a previously unreported pathway that included a new tactical combination or strategy that had been discovered by the computer. The synthesis began with an enantioselective addition of vinyl epoxide 1 to a protected vanillin 2 according to Creech's methodology. This was done in a relatively high yield and followed by a reduction of benzyl alcohol 3 after a trityl protection. The final step was olefin metathesis using second-generation Grubbs catalysts with commercially available styrene derivative. Cynthia's pathway was fully validated at the bench to successfully give Emperonine in a 27% overall yield and 97% enantiomeric excess for the S enantiomer. It is also worth it to note that this pathway was easily adjusted to use the opposite enantiomeric form of iridium catalyst in step 1 to easily adapt and synthesize the unnatural R Emperonine in a 28% overall yield and 98% EE. In conclusion, I hope today that I have been able to show you several examples of how Synthia can aid a scientist in their route design process by providing not only a customizable search setup to find the most relevant and important results according to your needs, but also to have the potential to be another tool in a chemist's toolbox, saving you time and money by accelerating your pathway design, finding pathways that come back to commercially available and known starting materials, and potentially even generating intellectual property in the process, ultimately getting to benchtop execution and your molecule in a bottle much more quickly. With that, I'd like to sincerely thank everybody for their time today, and I'd be happy to answer any questions within the Q&A period. Thank you.